Hi guys, today we're gonna be talking about one topic that I really had trouble with this quarter, relating to like eDNA. So for eDNA, I think I had most trouble with the concept of meta barcoding. In the initially, obviously the word is very long. I was a bit intimidated, but obviously I know right now that it means that it's like a method of or technique of analyzing eDNA, which is environmental data DNA, which comes from like shedding skin or like remains or like feces or whatever in the environment. So it could be in soil, water, air, whatever. And then it's broken down into multiple steps. So you collect the eDNA, and then you extract it as specific gene regions. So I had a lot of trouble with understanding how sequencing of DNA works and how certain uh, certain genes can be amplified, so you can study it and compare it to other genes, or like from one species to another species, or even from the same species, and comparing them to each other. So just I haven't had much experience with lab work, to be honest, and the like for example the week 4 assignment, I think it was on looking at a sequence of we used a online website, but we used a sequence for the DNA and then matched it with the corresponding organism. And I think I had trouble with that as well, just because I saw so many like uh, new terms, like just like the matching, like, and how like the accuracy or like the I had always assumed like it would be 100% match, but obviously there's like different experimental error, and then there's also like sometimes each organism will be different, like because of mutations and just genetic variation. But mostly that all that together, um, it just was a lot of information at once, I think. So I had a little bit of trouble grasping all of it at once. But I think breaking it down into steps, or like being able to understand that you just have the extraction, and then maybe like a PCR step where you're amplifying the amount of DNA, and then being able to take the DNA at specific regions, analyzing it, and then getting feedback from that. And using that feedback, we can do bio analysis, and you search up biodiversity, or like how many species are in given environment, or how many different species are in a given environment. This is all really important. And then I think another aspect that I had trouble with was understanding the different types of diversity. Like, for example, alpha versus beta diversity. That's another topic that I had trouble with. But overall, everything is very connected, or like all the topics that we learned in class are very connected. So from the beginning to the end, each step was building upon the knowledge from the previous class, and like the previous in-class assignments, and like the pre-quizzes and all those things. So I think that was really important for me. um Besides that, honestly, I think for like technology, there's like we didn't use much like complicated technology or like actual experimentation, but I think just being able to break it down was really important for me, and breaking down that each step of meta barcoding, and understanding that, just reading and understanding the connections between meta barcoding, eDNA, biodiversity, and also how bioinformatics can help um, us in so many ways, as understanding how organisms interact with each other, and what organisms are important for interacting with each other, and how biodiversity can impact the sustainability of a environment, how pollution and whatnot could damage various environments, especially with the marsh lab that we did. That was all very important, and being able to learn these step by step within each week and building upon it helped me have this break down the like preconception and the confusion behind meta barcoding just as a topic.